Hi, I'm Dr. Malaka El-Mufti, Professor of Microbiology. In this lecture, we'll discuss the bacterial cell structure. By the end of this lecture, students should be able to illustrate diagrams of gram-positive and gram-negative bacterial cell wall, to outline the differences between bacterial extracellular projecting structures, to discuss the passage of molecules through bacterial cytoplasmic membrane and through the outer membrane of gram-negative bacteria, to determine the virulence of bacterial cell capsule, to list the bacterial cell wall functions, and to distinguish between the bacterial cytoplasmic components and relate the component which could be a site of action of certain antibiotics. Now, the bacterial cell structure. The bacterial cell is a primitive cell. It is a simple cell, the so-called prokaryote. Pro is primitive, and karyote is in relation to the nucleus, the primitive nucleus. Whereas the mammalian cell is an eukaryote, a true cell with a true nuclear structure. Bacterial cell, or the prokaryote, is simple in every aspect and structure in comparison to the mammalian cell, except for one structure, the cell envelope. The cell envelope in bacterial cell is a much complicated structure. Now, bacteria, to be examined, we have to use the light microscope, and they are stained by the Gram staining technique. And bacteria are classified into gram-positive bacteria and gram-negative bacteria. The gram-positive bacteria, they stain violet. They take the color of the crystal violet stain in the Gram staining technique. While the gram-negative bacteria, they stain red. They take the red color of the safranin, the counter stain in the Gram staining technique. Now, the bacterial cell is composed of a protoplasm which is enclosed in the cell envelope. This is the cell envelope. The protoplasm is composed of the cytoplasm and intracytoplasmic structures that are to be discussed later, like the nucleoids, the ribosomes, inclusion granules, and the plasmids. And there are also some extracellular or extra cytoplasmic structures like the capsule, pili, and flagella. The capsule, the pili, and the flagella. We'll start by the cell wall. Cell wall is the layer just outside the cytoplasmic membrane in bacteria. The cell wall is a rigid structure with the so-called tensile strength. It has the power to be to be elastic. This is important because the pressure inside the bacterial cell is much higher than the atmospheric pressure. So the bacterial cell can lyse. And this cell wall, with its rigidity and tensile strength, can protect the bacteria from lysis, the so-called osmotic protection. And the structure that gives the cell wall this tensile strength is called the peptidoglycan. Peptidoglycan, which is a unique bacterial structure in bacterial cells only. This peptidoglycan in the cell wall of bacteria varies according whether the bacteria, we are discussing the gram-positive bacteria or gram-negative bacteria. In the gram-positive bacteria, it is a thick layer, more than 50% of the cell wall structure. Whereas in the gram-negative bacteria, it is a thin layer, from 5 to 10% of the cell wall structure. So remember, the peptidoglycan gives the cell wall its strength. Now discussing the gram-positive bacterial cell wall. We said that the bacterial cell wall has the so-called peptidoglycan, the component that gives the cell wall its strength. And in the gram-positive bacteria, this component will be more than 50% of the cell wall, thick layer. There are also other components in the gram-positive bacterial cell wall, the so-called teichoic acids. These teichoic acids, these are polymers of ribitol and glycerol, and they are antigenic. They form an antigen in the gram-positive cell wall, gram-positive bacteria. The main function of the teichoic acids is the stability and anchorage of the structures in the cell wall. As you see here, this is the teichoic acids, which is fixing the structures of the cell wall. It is antigenic. And also some sugars like mannose and other acidic sugars like glucuronic acid. Now, the structure of the gram-negative cell wall. Again, the cell wall has this tensile strength, this rigidity, which is given by the peptidoglycan layer. But in the gram-negative cell wall, this layer is a thin layer, about 5 to 10% of the thickness of the cell wall. You see here, it's a very thin layer. The gram-negative cell wall also has some important and abundant proteins. For example, the lipoproteins. Lipoproteins, these are abundant proteins which have an important function, which is the stability and anchorage of the cell wall. They are fixed to the peptidoglycan layer, and they also fixed to the outer layer. They give stability and anchorage of the structures, the lipoproteins. The gram-negative cell wall has also a unique structure, the so-called an outer membrane. This outer membrane has or is a two-layered structure. We say two leaflets. It has two leaflets, two layers. An inner layer or an inner leaflet, which is very similar to the cytoplasmic membrane structure. And what's important here is the outer leaflet, the outer layer, which is the so-called lipopolysaccharide layer. Lipopolysaccharide, so it has two parts. The lipid part, which is lipid A, it is the endotoxin of bacteria. And endotoxin is considered as one of the virulence factors in gram-negative bacteria. And it is released only upon bacterial cell lysis, only from gram-negative bacteria. And it can be also responsible for some systemic reactions of the infection. Now, the polysaccharide part, this is the outer part. It consists of a core or a stem of fixed parts and repeating units of the polysaccharides. These are interchanging, and these represent the somatic or the O antigen of gram-negative bacteria. The O antigen is a group antigen in gram-negative bacteria. So this is the outer membrane, or with its two leaflets, and the importance of the lipopolysaccharide layer. This layer has also some openings, the so-called pores. These pores are guarded or controlled by some proteins, the so-called pore proteins. They guard these openings, and these openings allow the passage of molecules inside and outside the bacterial cell. Only small-sized molecules, they can pass through these pores. The large-sized molecules cannot pass, and this can explain for some, as an example of some antibiotics that cannot pass to the inside of the bacterial cell, which is considered as one of the methods for resistance to the antibiotics. So the pores with the pore proteins. Now we can just say the functions of the cell wall from what we discussed later. Sorry. The cell wall gives the bacteria its shape, for the shape of the bacteria. With its tensile strength, it gives the bacteria the osmotic protection. It can protect bacteria from being lysed. And in the gram-positive bacteria, there is a teichoic acid, which is an antigen. Presence of antigen in gram-positive bacteria. And in gram-negative bacteria, we have the lipopolysaccharide layer, and the polysaccharide part is the O antigen. So there's an antigen, or the somatic antigen, in gram-negative bacteria. It can have some receptors for some antibiotics. The beta-lactam antibiotics, like penicillins and cephalosporins, they can bind to receptors in the peptidoglycan layer. And they could have some receptors for bacterial viruses, the so-called bacteriophages. And it can play a role in cell division. These are the main functions of the bacterial cell wall. And again, we see here the difference between gram-negative and gram-positive bacteria. And remember, the peptidoglycan layer is a thick layer in the gram-positive cell wall, gram-positive bacteria, the thin layer in the gram-negative bacteria. This table shows some differences between gram-positive and gram-negative bacteria. As regards the gram reaction, the gram-positive bacteria will stain violets. They take the color of the crystal violet, as we said before. And the gram-negative bacteria, they stain red, and they take the color of the safranin, which is the counter stain in the gram-negative bacteria. As regards the cell wall thickness and peptidoglycan layer, the gram-positive bacteria is much thicker, which with a thick abundant peptidoglycan, as I said, more than 50% of the wall thickness. Whereas in gram-negative bacteria, the wall is thin, with a thin peptidoglycan layer from 5 to 10% of the wall thickness. Teichoic acids, the antigen, in gram-positive bacteria only. The outer membrane in gram-negative bacteria only. The outer membrane with its two leaflets, you remember, the inner and the outer part. The porins, or the pores for passage of molecules, present in gram-negative bacteria only. The lipopolysaccharides in the gram-negative bacteria. Lipid part, the endotoxin, polysaccharides, which represent the O, or the somatic antigen. As regards to sensitivity to penicillin, which is one of the beta-lactam antibiotics, which is more in gram-positive bacteria than in gram-negative bacteria. Now we discuss the cytoplasmic membrane, or the cell membrane. This cytoplasmic membrane is a thin semi-fluid layer. It is a phospholipid bilayer. Phospholipid bilayer. Similar in structure to the inner leaflets of the gram-negative cell wall. It carries many functions, mainly the transfer of solutes and selective permeability. It has pores that allow the passage of molecules inside and outside bacterial cell. Passage of molecules inside, like the nutrients, and outside the cell, like the waste products. It also has enzymes like permeases, which allow or which help the entrance of molecules inside the bacterial cell. And also it can produce or secrete some enzymes, hydrolytic enzymes, that will degrade any large-sized molecules to make them in smaller parts, so that they're allowed to enter the bacterial cell. It also is responsible for the electron transport and oxidative phosphorylation. Actually, it takes the part of the mitochondria in the mammalian cell, because mitochondria are absent in the bacterial cell. The function is taken by the cytoplasmic, or the cell membrane. This diagram shows again differences between gram-positive and gram-negative bacterial cell, the thickness of the peptidoglycan here, and this is the cytoplasmic membrane, the so-called phospholipid bilayer. Now what about the intracytoplasmic structures, the internal structures? These are DNA, ribosomes, inclusion granules, and plasmids, and the nucleoid. The ribosomes are the site for protein synthesis in all cells. And ribosomes in bacterial cells are of a different configuration other than those in the mammalian cell. In the bacterial cells, the ribosomes are of the 70S configuration. 30S and 50S subunits. And they are sites for protein synthesis, and they can contain receptors for some antibiotics that act by inhibition of protein synthesis in bacteria. The sites for antibiotics that can act by inhibition of protein synthesis in some bacteria. Other intracytoplasmic structures, like the bacterial nucleoid. What is the nucleoid? The suffix oid means like structure. It is a nucleus-like structure. It's not a true nucleus. Remember we said the bacterial cells are prokaryotes, primitive cells. This structure lacks actually the nucleolus and lacks a nuclear membrane. So it is not a true nucleus. It is a nucleoid, nucleus-like structure. And the DNA are in the form of thin fibrils inside this nucleus. Thin fibrils of DNA. Another intracytoplasmic structure are the plasmids. What are plasmids? These are circular extra-chromosomal DNA parts. They can encode many traits for the bacteria. For example, they can carry genes for antibiotic resistance. The plasmids, the small circular extra-chromosomal parts of the DNA. And the fourth intracytoplasmic structure is the inclusion granules, storage granules. For example, these are granules or spaces that can store, for example, lipid, glycogen, starch for the bacteria. So these are the intracytoplasmic structures. The ribosomes, the nucleoid and the DNA, the plasmids, and the inclusion granules. Nuclear structure. These are the plasmids, the circular extrachromosomal parts of DNA. And this is bacteria with its intra and extracytoplasmic structures. Now we discuss the external structures, and these are present in some bacterial species, not in all bacteria. These external structures are the capsule, the outermost layer of the cell envelope. You see here the envelope is made of the capsule, cell wall, and cytoplasmic membrane. This consists the bacterial envelope. The outermost part is the capsule, which is present in only some bacteria, the encapsulated bacteria. It is a muco-gelatinous layer. It consists of polysaccharides, and these are antigenic, of different types of antigens. So the capsule is made of polysaccharides. In rare bacteria, capsules can be of proteins, polypeptide. But when you say bacterial capsule, it is made of polysaccharides and they are antigenic. The capsule is considered as one of the bacterial virulence factors, the factors that allow the bacteria to be pathogenic, because capsules can prevent the action of antibiotics to the bacteria, and at the same time, capsules can prevent the bacteria to be phagocytosed by macrophages. They can prevent opsonization. They protect the bacteria and prevent them from being phagocytosed. So the capsule is considered as one of the virulence factors. Now, these structures, other than the capsule, are some projecting structures from the external surface of bacteria, like the flagella and the pili. What are the flagella? These are the organs for bacterial locomotion, for bacterial motility. And they are filamentous but rigid structures made of a protein called flagellin, and this protein is antigenic. It represents the H antigen in motile bacteria. It is antigenic. And these flagella are actually firmly attached to the inside of the bacterial cell, like a plant which has roots and the outer part. The flagella are also fixed firmly to the inside of the bacterial cell, and they project to the outside. And they have different distribution. They can be either at one pole, single, or any flagellae at one or two poles. And they can be all around the bacterial cell. Monotrichous, mono means one. Trichous means the hairy projections or two projections. Lophotrichous, a bunch of flagellae at one pole, or peritrichous, flagellae all around the bacterial cell. And remember, they are the organs for bacterial motility, for bacterial locomotion. And they are the sites, or they represent the H antigen in motile bacteria. The other projecting structure are the pili or fimbri. These are thin, hairy projections outside the bacterial cell, and they are formed of a protein called pyelin, and they are short, straight. They're not firmly attached to the bacterial cell, to the inside of the bacterial cell, and they are of two types: the common pili and the sex pili. The common pili are responsible for attachment. They have certain endings, adhesive endings, that allow the adherence of bacterial cell to mucous membranes. And this is considered as a virulence factor, a factor that allows disease production. Once they are adherent to the mucous membrane, they cannot be washed by secretions. They stay and they can start the pathogenic process. The other type of pili, the so-called sex pili, they play a role in one of the methods of gene transfer in bacteria, which is called conjugation. They form channels that allow the passage of genetic material from a donor bacterial cell to a recipient bacterial cell. And these channels are the sex pili for passage of genetic material from donor to recipient cell in the process called conjugation. And these are the extracellular structures. Thank you.